while that is warming up, let me just introduce Rob Tibshirani. He's a professor of statistics here at Stanford and worked a couple years back with a postdoc from Mark Davis's lab, Shai Shenor, on a way to basically deconvolute the signals from mixed tissues like blood to get cell-specific gene expression signatures, and so that's the work he's going to talk about today.
Thanks, Holden. Hi, everyone. As mentioned, this is work with Shai Shenor. It's actually continuing. He's now has his own lab in Tel Aviv, but we're continuing this work. But this was done when he was here as a postdoc. And as Holden mentioned, I'm a professor here. I'm not a biologist. I'm a statistician. I'm a professor here in the Department of Statistics and Health Research and Policy. So I work with a lot of people in the med school and in biological sciences, but I'm not a biologist by any means, as you'll see. And I've given this talk to this course in the last couple of years. Same talk, same joke. So in the next slide. But I think the joke still applies. So if you, it's to do with the challenge in communication between statisticians and biologists. A statistician and a biologist are about to be executed. Each one's granted one last wish. The statistician asks that he be allowed to give one last lecture on his grand theory of statistics, at which point the biologist says he wants to be executed first. That's his wish. Although the update is that it's not so bad being a statistician these days. Actually, we're more sexy, apparently, according to Hal Varian of Google. But seriously, it's a really exciting time to be a statistician because there's lots of data everywhere, right? Of course, in computational biology, which you guys do, but also on the web and in finance, there's a huge amount of large data sets being produced, and the challenge of how to find the signal among the noise is really great. And so this field is actually pretty young, statistics. Like, it grew up in sort of a small data world until about 1970, and now with the huge sets of data which are being produced in various fields, the challenge of how to deal with this is really it's new. And it's not like the old methods can just be recycled. As you'll see, we need some new techniques, and I'll just talk about one today which is of this flavor. So the main driver of this work is Shai Shenor, who was a postdoc here at Mark Davis's lab a few years ago, and now a professor in Israel, and a lot of co-authors who I mentioned here. And this paper was published a few years ago in Nature Medicine, I'm going to talk about. So, by way of background, what problem I'm going to talk about, gene expression arrays offer a means, of course, of quantifying the gene expression of potentially the entire genome in a single tissue, right? So we take a sample somehow of solid tumor or blood from some tissue, put it on an array, and we get the gene expression, the RNA levels of potentially all the genes in the genome. Okay, so it's a very powerful technique. It was developed partly here at Stanford by Pat Brown in his lab, and is now used, of course, around the world, and it's become quite cheap to use, and it's very popular and a common tool. And one thing it's used for quite often is to compare two groups, right? So we have a control group and a disease group. We want to know which genes have higher or lower expression in the disease group versus the control group, right? So that's a very common use of microarrays. And in statistical terminology, that becomes a large number of t-tests, right? We have two samples we want to compare across a large number of parameters, namely the gene expression of each of maybe tens of thousands of genes. So we want to do maybe 10,000 t-tests, compare one group to the other group. And this actually has brought quite a flurry of activity in the statistics world, and there's been a lot of methods and books and software written about this problem in statistics. Our lab produced the SAM method, symphony analysis of microarrays, which is a commonly used technique for this problem, for comparing genes across two conditions or more than two conditions. Okay? So this is a commonly used problem, a commonly occurring problem, and there's lots of methods of approaching it. Okay? Basically, it's t-tests, plus you have to worry about the fact that you're doing a lot of t-tests. So things like false discovery rate become important. Right? You can't just look at one. You can't look at t-tests in isolation. You have to account for the fact you're looking at tens of thousands of tests and worry about how many false positives are you likely to see. So things like false discovery rate become important criteria. Well, what's one problem with this technology is that often the tissues that give you gene expression are not homogeneous, right? they're a mix of different cell types. So what you're getting is a soup of different cell types, and the gene expression measurements are typically a measurement of that whole soup. So what you're getting is not necessarily the gene expression of one cell type, but the entire soup. Now, there are methods coming where you can do gene expression on single cells, but they're still not really scalable yet and cheap enough to be used commonly. So we're looking at a bulk group of cells. What you're getting is a mixture of cell types when you measure gene expression. You're not sure, for example, if the gene expression is high, is it because the actual cells have changed in the disease sample, or you just have more of one cell type than another? Right? You can't tell which or both. So there's a confounding between the proportion of cell types in the disease sample and the gene expression of the genes. So one solution is to just ignore that. And that's what basically we've all done for now, is to say, well, I've got the heterogeneity, but I'll just do the bulk measurement and hope that I can see sort of gross effects, ignoring the fact that there's heterogeneity there. Okay. The other approach is to actually separate the cells into different cell types and measure the gene expression on the separated cells. The problem with that, at least Shai tells me, is that it's for one thing, it's expensive. It may also affect the expression. The act of removing cell types from the other surrounding cell types can alter the gene expression. So you've changed the actual system you're trying to measure. You can just say, well, it's a power problem, let's get more samples. Well, that's only going to work if there's no systematic differences between the cell frequencies. So we have this confounding between proportions of cell types and gene expression, and we want to somehow untangle that confounding. So what I want to talk about today is a simple idea is to get some more information and then use that information to deconvolve the gene expression. So we're going to count the proportion of each cell type in each sample using, for example, FACS. Right? So we're worried that the cell type proportions are the same, so we'll count the number of cells in each cell type in the sample, and then use this information along with the gene expression for the total sample to deconvolve and hence get an idea of the gene expression for the pure cell types. Right? So we like to separate the gene expression, the cell types physically, but for the reasons I just said, that's not practical or changes things. So we're going to do it in silico, essentially, by doing this deconvolution. And the first time people see this, it looks like black magic. It looks like, well, you shouldn't be able to do this, but I'll hopefully try to convince you with simple examples that it's actually pretty straightforward how this works. Okay? So I've just got a couple of equations. This slide and then a picture on the next slide will show the basic model. So suppose we have gene expression values I'll call xij for sample I1 through n and genes 1 through p. So n might be a few hundred samples from patients, and it might have 10,000 genes from a chip microarray. Okay, so this is what I measure, and this is what I would normally just use this data and do like t-tests or however I want to make comparisons of the gene expression across conditions. But now I'm going to measure this additional information, the cell type proportions, okay, using, say, FACS. So for each sample, I'm also going to get, for each of k cell types, and in the examples you'll see k is maybe 2 or 5, okay, the proportions of the cells in each cell type in each sample. Okay? And now I'm going to assume that what I'm seeing is a mixture. In other words, so the x is observed, and the w is observed. So I observe this x. I'm going to assume that the gene expression I'm seeing for sample I, gene j, is a mixture of these pure gene expressions in different cell types. And these guys I don't see. Right? These are what I want to recover. So I'm assuming what I'm seeing is a mixture, like a soup, of these pure components, unknown, mixed by these cell types in proportion to these cell type proportions, which I have measured. So this is observed, and this is what I want to uncover, and then I have to have some errors around that mean, okay, which I'll assume that means 0. So this is why it's called a deconvolution problem. We observe this, we want to back out this information from the total gene expression and the cell proportions, cell count proportions. So in pictures, this is what I observe the gene expression here. Usually these are drawn the other way, and you have genes, but in statistics people like put the samples in the rows. So I observe the gene expression for n samples and p genes. I also observe the counts of the cell types from using FACS for each of these cells. So again, this might be Might be 5 columns here. And this is what I want to know. This is the pure gene expression, the gene expression for pure cell types, which I don't get to see physically. So basically, statistically now, it's the procedure is simple now. I do a regression of each column here on these cell types to get out a column of H. So it's just least squares regression, right? Which is sort of, this is just a linear model. This just says that this observed guy is a linear combination of these proportions, and these weights are what I want to know, these H's, and I just get them by regression. So I do a linear regression of each sample, the gene expression of each gene for all samples on this, the measured cell proportions to give me a column, the gene expression, this gene, for across the cell types. I do that for each different column. So I have to do a total of p different regressions, each one yielding one column of H. Yes?
So here there's an assumption that you have equal expression throughout your different populations, right?
Equal expression.
Like you don't have, if you have enrichment in gene expression in one cell type,
Yeah.
then you can't really account for that, right? Because you're not normalizing.
No, I'm going to get.
Like,
Yeah.
are those cells sorted, or you just have an understanding of what the proportions are?
That's all I have right now. But I'm going to get from this. I'm going to get if they're enriched in one cell type, I'm going to learn that from this matrix over here. So at the end of all this, each row of this is going to be the gene expression for one pure cell type. And if there's enrichment in one cell type, then I'll see it by looking at the corresponding row of this matrix.
So the cells are sorted in this.
They're not sorted, no. No. Okay. It doesn't feel like I answered your question, but.
Are you asking about whether there is an assumption here, I guess, that the expression level of a gene in a given cell type is the same across the samples, at least for a given group?
Oh, was that your question? Yes. I'm sorry. Yes, and that is the case. Yes. So for a given cell type, I assume across my patients that gene expression, there's a common, that is the same. So there's a common gene expression for a pure cell type that's going to apply to all patients in the group.
There's a problem with that assumption.
Why?
You can't account for the heterogeneity.
Well, there'll be some heterogeneity. Well, I haven't got to about, for example, when I compare normal to disease, I'm going to apply this model within normals and within disease. I'm allowed them to vary. Now, you're right, there's more heterogeneity, of course, beyond that, but this is just an approximation to try to get out the gross signals, right? Now, compare that to these, if we didn't even have these cell types, we're making approximations even greater, right? That across all cell types, all patients, the gene expression is the same, which is, so we're moving down one more level. At least we're allowing the gene expression to vary across cell types. Okay? So hopefully this will make it clear. Again, this either is obvious to you or it looks like some kind of strange magic. So here's just a little toy example to help explain it. So I observed these three samples, and the gene expression. Here are and I observe these cell proportions, 0.9 and 0.1 and 0.1 and 0.9, and I want to know these guys. So I want to know the pure gene expression for, I labeled this wrong, sorry. The samples are on the rows here, that's why and these are the genes or the columns, as they were in the previous slide. So I want to know these guys given this information, right? So I do a regression of each of these guys on each of these guys to give these weights, and when I do that, I get this. Okay, so it's sort of like you get this soup, and I tell you overall the expression of each gene in the soup is given by this. Each sample's a soup. But I also tell you, by the way, there's 0.9 of one cell type and 0.1 of the other cell type in there. So you can use that information to untangle what the gene expression of the individual components of that soup are that would needed to make up this total, right? So that's the idea. Now, important point is here that the reason this works and the reason the method works in general, you need these proportions to vary across different samples, right? If everybody looks the same, if I have, say, 0.5, 0.5 in every sample, I don't have enough information to do the deconvolution. So it's depending on the fact that the mix of cell types across samples it varies, right? Which in this little example, I made it vary quite greatly, 0.9, 0.1, 0.1, 0.9, right? if all the samples look the same, they're all, say, 0.8 and 0.2 of one cell type and the other, I don't have enough information to figure out the gene expression that would have given the total. Right? You need to have some variation in the cell type counts in order to do this deconvolution.
But how large must that variation be? Like, for example,
Yeah.
if we're talking about immune system and we're saying, I don't know, 10% B cells in bone
Yeah.
marrow,
Yes.
and it's roughly approximately 10% for all
Yeah.
the patients or all the
Right.
normal controls in one group. How
Yeah.
much does that have to vary for you to work your statistical magic?
Well, of course, it depends, and at the end of this, you get also an idea of the standard error of these numbers, so that's going to be reflected in that. But you need, so it depends. It depends also on how much these guys vary. So, if these are very, very different, then a small variation here will be enough. If these guys are pretty close, if the pure gene expressions aren't very different, you're going to need much more variation here to untangle them. So, yes.
How do you know how many different cell types there are?
Well, you don't. Basically, so I think that's. I think my understanding is you sort of you make a guess at the major ones. Like here, when we see example, we'll look at 5 different cell types in blood. Of course, there are many, many more subtypes. So you basically try to make a good guess of what the important ones are, and you can drill down as far as you want, and your information will sort of get less and less as you go farther down. Yes.
I'm wondering if this
Yeah.
might be similar to the question Sarah was asking. So for instance, if your cell type distribution is 10% B cells and 90% something else, but the B cells are making 90% of the gene that you're looking at, and all the other cells
Um,
are only 10%.
So you say they're making 90%, you mean of the gene expression? They're making 90% of the gene expression?
For instance,
Yes.
so the 10%
Yes.
of the cell bulk,
Yes.
but actually they're
Yes.
responsible for 90% of
Yeah.
the gene.
The total, yes, right.
Is that
Yeah, that comes out here. So what you're getting here, no, it's not lost. So what you're getting here is the pure gene expression of one cell type, and then so that's accounted for here. So in this, so if the proportion, sorry, you're saying the proportion of B cells and T cells is different, but
say
yes,
cell type
right,
2
yes,
in the top row, which represents 0.1 of the total cells,
yeah,
actually is responsible for 90% of your
Yeah.
gene expression.
right. So then you see big numbers here. You see big numbers in this guy here because this is the pure gene. This is the idea of this is the pure gene expression for that cell type.
So you're
Which-
not just assigning 10% of the total gene
No.
expression to that cell type, which is representing
Right.
10%.
It's if you had one cell from that cell type. This is what the gene expression would be, and then it's mixed together with these proportions. So this is exactly trying to get at what you're saying. If the gene expression is higher in a pure cell type, you would see it in this guy in the corresponding numbers here.
How do you know which cell types it is? Do you look at other markers as well that would be?
Which cell? Well, the information is just coming from here, right? It's saying, I mean, imagine you're seeing. So imagine the situation you're saying is right, that there's the gene expression is higher in B cells versus T cells, right? Then if you see enough people with some with a low percentage of T cells and some with a high percentage of T cells and B cells, right? So the mix is different. You can untangle that because you'll see the people that have higher proportion of B cells will also have higher gene expression. So you're teasing that information out by looking across different people, all of whom, gets back to the earlier question, we're assuming that each person's gene expression for the pure cell type is the same across different people. So you and I, we all have the same.
Within the
But
same person,
within this-
there's different,
Yeah.
your B cells, for example,
Oh.
are accounting for 90% of your transcripts.
Yes.
Say your T cells are
Yeah.
10%
Yes.
of the transcripts of a specific gene.
Yes,
Then how do you account for that?
that's what this measures, these guys, exactly. That's what these guys measure.
It's the magic.
Yeah. Well, let me push on and I can maybe ask more at the end. I'm not sure if okay. So, how do we use this? Well, we use this by, say, if we're doing a 2-group comparison, normal versus disease, we apply this deconvolution within each group. And now we can compare, rather than compare the total gene expression, we compare the pure gene expression. So, I mean, again, the idea is that if we could separate the cells and get the gene expression for pure cell types, we would do it just physically, and we start with this data. But rather, we're doing this in silico, and now we imagine having done this that these are the pure gene expression for each of the cell types, and now we can use these, say, to compare normal versus disease within each cell type. Okay, using, say, a t-test or SAM. We can also create adjusted data that which basically is what we'd expect to see if each of the proportions was the same. So every person has the proportion of cell types is different. mean, that's what we're relying on that fact to do this, right? Each person's soup is different. The relative proportion is different. Now we can ask the question, given the estimates of the pure gene expression, what would the total gene expression look like for everybody if we all had the same proportions, right? So we can back that out of the modeling. We can adjust the data to say this is what the data would look like for you if you had half T cells and half B cells. If everybody had half and half, this is what it would look like. So we can either do a bi-cell type comparison or a total comparison having adjusted out the cell type proportions. So let me show you two examples which hopefully will help to explain it further. So the first example is artificial, in which we took pure cell types and then mixed them together in various proportions. So here we knew the correct answer. So this is not a real experiment in the sense that we don't normally have this information, right? I started off by saying we'd like to separate the cells and measure pure gene expression, but that's not either feasible or it's too costly. But here's just a sanity check to make sure that our method works in a situation where we know the right answer. So we start with pure gene expression. We mix samples together in various proportions. We give that total data to the procedure, and we ask how well, we look to see how well does it recover the pure gene expression, which we know in this case, because we started with pure samples, pure cell types, and we mix them together. And then a real example where we didn't have this pure information, but we still want to tease out this different cell type gene expression and figure out whether there's a difference between the groups, which in this case was kidney transplant patients, stable versus rejection, with 5 different cell types from blood. So the first example where we mixed these two cell types in various proportions. Again, these proportions vary greatly, which helps the procedure. Again, if they're all the same proportions, we wouldn't get off the ground. Okay? So we have to have variability across the proportions, which we've arranged for here. So here's the results. Each of these points is a gene. This is for liver and brain. This is the measured expression for the genes in the pure cell type, liver. Right? So in this case, we take the pure cell type, we put that on an array. That gives us the gold standard. But now we also give the mixed information to the procedure, and we ask it to deconvolve, and that gives us the y-axis. And we can see we have very good correlation between the gene expression that the procedure tells us by deconvolution versus the measured. There's some tens of thousands of points here, so there is some kind of systematic bias, but it's a very small proportion of the genes. We didn't actually never got to the bottom of why this was the case in this example. And here as well, when there's errors, they seem to be all on this side. But again, the grand majority of genes are lying very close to the true values as measured by the direct gene expression measurement. So these are the pure gene expressions. And then if you ask, well, how does the total, if I then take the pure gene expression from deconvolution and put it back together in the proportion, what does the total look like? And here's that. So here's for a sample which is 75-25. Here's the total gene expression that's measured in a direct way. Here's what the model says it should be. And again, you've got good correlation in all the cases with this one-sided bias in some relatively small number of genes. Okay? So that's the case where we knew the truth, and we compare the truth to our predictions. Here's a case, a real example, which was 2 sets of patients having kidney transplants, and these transplants are either successful, so they're stable, or at some point, fairly soon after the transplant, the kidney is rejected. And Nini Sarwal, a nephrologist here, wanted to know, can we predict ahead of time which patients are going to have trouble with the transplant from the gene expression from blood? So Shai did CBCs for 5 different cell types for these patients. So we now have, by fax, we got the relative proportion of these 5 kinds of cell types from blood. And this is a case when if you just ignore the cell types and just do a t-test across the 2 groups, again, stable versus rejection, you see very little. So this shows the false discovery rate if I basically apply SAM. So I do t-tests across, for each gene, I compare the stable to the rejected. And the false discovery rate overall is about 60%. So there's very little signal if I just compare genes across the conditions. Either overall, or if I look at ones which either go up or down, There's very little signal. If I do a clustering of the two groups, stable and versus rejected, these twos, the twos and the ones are the two groups. This is I do hierarchical clustering using all the genes, and there's about 7,000 genes here, I think. You see very little separation. So there's no clean separation based on gene expression between the two groups. Okay, so hence we're trying to get deconvolution to help us out here, to separate out the gene expression of pure cell types and hopefully find something, some more structure here. So this is the procedure I just described. Again, and now it's just in graphical form. I've got my disease patients. Total gene expression for them from blood. The control, so disease and control here, that means stable and rejected, these two groups. I do this deconvolution, which we, by the way, call CS-SAM, cell type specific SAM. And there's the last slide, I'll show you some software. I mean, I'll give a link to some software if you want to try this on your own. We do the deconvolution, now we get we're going to get gene expression for each of the 5 cell types, right? For each of the two groups. So now we can ask the question, well, these two groups don't seem to be very different overall, as I just showed, but somehow maybe within the cell types I can find a difference between the groups that's going to be useful. So that does turn out to be the case. What we find that is in monocytes, so this is number of genes, this is the false discovery rate. There seems to be a fairly strong difference in monocytes. Okay? So what I've done here now, I've done the deconvolution. Now for each sample I have what I think is the gene expression for their monocytes. And that's what the models is giving us. And now if I compare those across the two groups, I get pretty strong signal. So it looks like there's a difference in gene expression in maybe 50 genes comparing stable to rejection in monocytes, which was there in the original data, but I couldn't see it because the different cell types were confounding it, right? Because I wasn't looking at the pure monocytes, I was looking at these 5 different cell types, and the proportions of different patients was different, so that was confounding the comparison. But now by deconvolving and pulling out the gene expression for each of the individual cell types, I get a cleaner look at the difference between the two. Okay? And yes?
What would have been your prediction just looking at the cell type proportions between those?
The prediction of what?
Whether they were rejecting or
A good question. There was no we tried that, and there wasn't a systematic difference in the cell type. So it's not like it's just more monocytes or more of something else. There was a mix. And it seems to be not the proportion of cell types, but actually the expression of those cell types. Yes?
How do you account for the clinical heterogeneity that may be aligning with the percentage of cell types? So it may not be that proportion of cell type that's determining your outcomes of the gene expression, but it's there's some clinical variable that is aligning with the percentage of the monocytes.
Now remember, this is not the percentage of monotypes. So we're not, conclusion here is not that the percentage of monotypes is it's the effect. It's the gene expression of the monocytes.
Well,
It's not, so we factored out the proportion of monocytes. That's not a factor now. We're saying if you just look at the gene expression of monocytes in stable and in reject, then there's a strong difference in about 50 genes. And not to do with the proportion of such cells, but the actual gene expression of the cells.
but you're determining the gene expression of the cell,
Yes?
assuming that a certain portion of that variation is going to be attributed to the proportion of that cell.
I didn't understand that statement. Could you repeat or rephrase? I didn't understand what you said.
With any clinical study with linear regression, you have to do multiple linear regression
Yeah.
models to determine the best fit based on all the confounding variables.
Oh, so, yeah. I mean, if what you're saying is there could be confounding variables that are causing this effect that actually are, so they correlate with monocytes, maybe monocyte gene expression or proportion of monocytes. You're absolutely right. There's always there could be confounding variables which we don't know about because it's not a randomized study. Fair enough, and that's. I'm not sure what to do about that here, except maybe to try to measure some of those and see whether, when you adjust for them, the effect goes away. Yeah.
Or just one easy way to confirm that what you're seeing is actually in the monocytes, then you could just go back and sort for monocytes and look at those.
Yeah.
And not have to do the entire experiment, but just
Right.
have that as your
And that's being done in some follow-up work, right? So, and if you look more closely, it's actually genes that have higher monocyte expression that seem to be the effect here. And If you then use the gene expression of monocytes to do the clustering, remember I showed you before that when I used total gene expression, the clustering was very ineffective. Now you get basically perfect separation between the stable group and the rejection. Oh, sorry, stables here, I think, and rejections here. So the information's the distinction between the rejection and stable seems to be in the monocytes, which we couldn't see from the overall comparison that didn't adjust for this the cell proportions. So this work's continuing with Pravesh Khatri, who's now just got an appointment here at Stanford, and Shai are working on this more to see whether this is actually a real effect. So I hope I've, to some extent, convinced you that this is a useful way of analyzing gene expression from mixed samples. The method, the technology that's published in Nature is called CSSAM, self-specific SAM. There's actually a package on the CRAN library in R, so the public library, the public repository, called CSSAM, which does this, and you're welcome to try it. And if you have, I say available from Shai, but he's since put it on CRAN, so it's public available in the R archive if you want to try it out. So that's it, and happy to take more questions.
Can I just make a comment?
Yeah.
Maybe from a bit more of a biological point of view. You know, what Rob was getting at sort of in the beginning here, and I think it's important, is this idea that when you look at blood microarrays and you look at, let's say, disease versus normal, you have a couple of possible ways that you can get different signatures. In one case, and a good example of this is like the interferon alpha signature that people see in lupus that's been described by a few different groups. It's largely an effect of differing proportions of cell types. Okay? Largely, maybe not fully, but mostly. Now, in that case, you're using the microarray as a really fancy cell count.
Right.
That's essentially what you're doing. But it's also possible, like in that kidney transplant example, where on average the cell counts of different cell types are not changing overall across your groups, disease and healthy, but the gene expression within them is. So, for example, the monocytes are getting activated or differentiated in some way, and that's the key difference. This method is for pulling out those kinds of cases.
So that's a good explanation. And as someone asked, in this case, the proportion of cell types was not different in the two samples. It's in the two conditions. But the thing that seemed to be different was the actual expression in the cell types of monocytes.